The title, our group is interested in lipid-protein interactions, and just as Benoit said, I'm going to pick two examples of the work that sort of related to X-ray scattering to be presented to this audience. Okay, so this gives you a sense of the problems that our group is interested in. We have a long history of looking at lung surfactant, and we delve into sort of the immunity through both antimicrobial peptide and specific peripheral binding protein in terms of response. And we have other lipid interactions that we're interested in terms of its interacting with polymer as a sealant, or what is happening when it is interacting with a very high curvature system. So today I'm going to pick two of these to discuss. One has to do with a intrinsically disordered protein, alpha-Synuclein, and trying to understand what its mode of interactions with the lipids are and what it is the physiological function resulting from that action. And the other one has to do with a family of protein, and which would be just sort of a proof of principle to show you a set of tools that we have developed and put in place to allow us to get at high resolution information in terms of what is the configuration of the protein when bound to a lipid surface. So I would sort of start by using this slide right here. You see, I hope you see my cursor, the lipid bilayer in a lamella flat type of format that is there. But its interaction with various protein can give rise to change in the curvature of the system. So in a case, even without any protein, just based on the spontaneous curvature, due to the spontaneous curvature of the lipid that is involved, that can give rise to a curved system, or interaction with transmembrane protein can actually give rise to curve either in the aggregated state or just because of the intrinsic curvature of the protein shape itself. And its interaction with the cytoskeleton would alter the shape of the local membrane. And in the case that we're interested in today, we talk about what happened if you have a peripheral binding protein inserting into the lipid that would also change the curvature. So this is just sort of a level setting for what can happen when it is interacting with a protein. So a set of proteins that is binding peripherally to the membrane can actually probe the curvature. So what is shown here is a cartoon of the membrane that is curved, and it has sort of, some exposure of the hydrophobic portion of the tail portion that is out there. And if I have a amphiphilic protein that actually have its hydrophobic side on one side and the hydrophilic residues on the other side, it would seek to insert itself into the membrane. And what is shown here in this review by Anthony is that as you increases the radius, so decreasing the curvature of the membrane, you see a decrease in the bound percentage of protein into the system. So this seems to suggest that it is really seeking for the curvature of the surface to which it seeks to bind. And so I want to start this by talking about a particular protein that we have been focusing on. And this protein has been implicated in Parkinson disease. And this is the alpha-Synuclein protein. So this is a state micrograph of a cell that you have the nucleus that is here. And you see what is called the Lewy body, which is one of the hallmark of the Parkinson disease due to some aggregated state of conglomerate in the system. So if I were to sort of look at the location of where does the alpha-Synuclein reside, and how is its interaction with the materials that is residing, co-localizing at that location. What you can see is that this happened at the synapse and at the synaptic junction that is here. So there's a lot of lipid species that is there, but there's a set of vesicle called the synaptic vesicles, which is highly regulated in terms of its size, and they are on the order of 40 nanometers in size. And what you can see here in this cartoon is that it seems that alpha-Synuclein is specifically seeking for these highly curved membrane surfaces for its binding. And so this is a stained micrograph that's showing the colocalization of the alpha-Synuclein with these synaptic vesicle. So what is alpha-Synuclein? I use the term of intrinsically disordered protein. So unlike proteins that we learn, from the textbook that has their secondary structure that is well-defined, the intrinsically disordered protein is by definition or by its name, intrinsically disordered. So this is alpha-Synuclein that is in the bulk, and this is empty simulation that actually shows that when this intrinsically disordered protein is bound to the surface of that membrane, it actually undergoes a conformational change whereby it turns into this empipathical alpha helix that started from the N-terminus, and it would sort of embed itself into the membrane. But even when it is fully associated with the membrane, it seems that the C-terminus, which is the green part that is here, still remain intrinsically disordered. So you could imagine that there's one part that is anchored to the membrane, and then the other part that is just sort of flopping on the surface of the membrane, as shown in this simulation. So the question of how alpha-Synuclein actually probes the curvature of liposome has been studied by other groups. So this is Liz Rhodes' result using vesicles of different size to really look at the dissociation, constant of these vesicles, as well as showing, this is very sensitive, I apologize. Okay, I won't touch. And also showing, the molar partition coefficients of that. And so this gives us a sense of the sensitivity. But as you know, when we're making lipid vesicle, when we are trying to control a very precise size of this vesicle, it becomes rather difficult. And so what is shown here is that, if I use the extrusion method, the liposome formation gives rise to really a polydispersed system. So, in our lab, we usually go through some sort of, filter with pore size of a certain size, and we'll look at the distribution. And when you're looking at things that are on the order of, large hundreds of nanometers, 100 nanometers on the size scale, then your polydispersity would fall into the range that you can tolerate. But if we were trying to really distinguish the association to try to get at this spontaneous curvature problem between something that is 40 nanometers in diameter versus 50 versus 60, then the polydispersity would give rise to a pretty unsurmountable hurdle for us to definitively call on the curvature sensitivity of this system. So there has been strategy that has been put in place in order to really get around this polydispersity. So this is a method that is by the Stachowiak's group that actually look at a polydispersed sample of adsorbed vesicle on a surface, but by picking the particular focal plane that you look at, you select out a certain size on which you are doing your analysis of this overall polydispersed system. So that's one way to get at this. But if we want to get structural information as well as binding information, this would be a little bit difficult to achieve. So what we thought of is, why don't we combine the technology of having very robust monodispersed nanoparticle size and use that as a template to form a lipid bilayer. And since we're looking at peripheral binding protein that does not involve the penetration onto the bilayer, this would be a good method to help us to have a monodispersed size distribution of the entity that we're looking at. So it is monodispersed in terms of size and shape. And we can do these bulk measurement whereby we can look at the binding affinity and bound fraction. And these systems are also stable. However, there are limitations with this technique. So what is shown here are silica particle that is used, and then we're trying to form a lipid bilayer that coats the particle. So and you can form this bilayer as shown in this micrograph that is on the right-hand side. However, there are limitations as to how much you can charge that lipid to still be able to successfully coat the surface. What you want is to be able to coat it completely so that you don't expose the silica surface to be interacting with the environment. And if that were the case, then you would be sort of contaminating the data that you are looking at. So if I were to look at a relatively larger size of diameter of something that is over 100 nanometers, the maximum amount of charged lipids that I can put because of the repulsive interactions of the charge-charge interaction would be around 20% of charge. But if I want to scale it down to this length scale that we're interested in to mimic the synaptic vesicle, then it would even have to go with a lower charge. The problem, however, is that, in the system that we're looking at, we need to go higher than what this particular system would allow us to sort of naturally forming. So in a way, what we want to look at in order to really to coat it, because, it costs energy to bend the system, but it also you have to adhere first before you can break it up to coat the system. So the question is, how can you effectively form a overall full coverage of lipid bilayer that is coating this nanoparticle that has very high curvature? Okay, so it turns out that we would need to address the previous two forces that are at hand, or the two energies that has to be taken into account by using a two-step process. If I have the silica, first I would like the vesicle to really adhere to it. And after the adhesion, we want it to be able to be on site, broken up to rupture so that it would coat the whole silica particle that is there. And so in order to increase the adhesion, we play around with the surface chemistry. And so, by surface functionalizing the silica particle, by OH or by NH2, we were able to alter the surface behavior to allow us to have an adhesion. So I have the particle and I enhance the interaction of the vesicle so that the on rate and the off rate can be adjusted so that it would adhere to the surface. And you can clearly see the adhesion once I make the modification of the vesicle onto this chemically modified surface. However, the fact that it adhered to the surface doesn't mean that it would break open. And it is clearly seen in the previous picture as well as this, which is they would adhere, but they would also interact with the other particles, forming an aggregate, which is not what we want. And so the second step of how we create this is really to use osmotic stress to stress the system so that it would rupture. And so the first trial, we sort of look at using glycerol as the osmolyte to allow us to open it up. And when we pump it up to a correct concentration, what we see is that we can robustly give rise to these coated with a single layer of bilayer of nanoparticle. And so the size scale that you're seeing here is around 70 nanometers because these are 60 nanometers in diameter nanoparticles that is coated with a bilayer, which is 5 nanometer on each side when you look at the cross section of this. So we call this SSLB, standing for a spherical nanoparticle supported lipid bilayer. And this just sort of, to go through the whole sort of process at which we make this particle. And as you can see that this is sort of in a one test tube type of configuration that we can make it, we can add it, we can shock it, and then it would be coated. And then we can centrifuge out the coated SSLB so that we can put it into our study. So what we have looked at is using different type of osmolyte, and depending on which one you use, you can find the threshold value at which this coating would occur. And so this is with sucrose on the top layer, and the bottom layer is with glycerol. And you can see the same thing. Once it goes to a certain osmotic stress, you get the system, it helped to destabilize and burst it and rupture it to form this bilayer onto the surface. We have also then dialed down from the 60 nanometer diameter size of the silica molecule all the way down to the 40 nanometers. And as you can see, we are actually increasing the loading of the charged lipid, DOPA, in a milieu of a spheroionic lipid of DOPC. And you can see that even with 50% as opposed to the 20%, which is the former limit, when you don't use these other tricks of the formation of the system. Similarly, it's irrespective of the lipid that you use, the charged lipid that you use, we can also achieve similar results with changing the head group from PA to PS. So what this shows us is that we have a robust way to give rise to a monodispersed population of very small particles, small in relationship to what we are trying to look at, that would be a mimic for the synaptic vesicle to look at its affinity, the affinity of alpha-Synuclein to these different states. And these can be highly curved and highly charged. But what is important is because it has the silica as the substrate, it also has very high scattering cross-section, and we'll get to that part of it. And so to ensure that this particular system actually is a robust system that doesn't really interfere with the absorption of the alpha-Synuclein to the surface, we first did isothermal titration calorimetry, whereby we want to dial in and see how does the protein actually interact with this SSLB that we have found. And so these are just sort of, the heat curve that you would see, and that translate to, as a function of the increasing mole ratio, how the injection, how the kcal per injection looks like. So if I were to compare the vesicle work that we got from the literature that has some polydispersity and hence a larger error bar that you can see here that is plotting the lipid to protein ratio as a function of a curvature, looking at the binding density of these materials on the surface of the lipid, either lipid itself or lipid-coated surface, you see that the SSLB actually increases the alpha-Synuclein binding density on the surface, and also, as seen by the error bar, really, because of the monodispersity, give rise to a tighter control of delineating what this surface binding density is. Likewise, SSLB also increases the alpha-Synuclein binding affinity compared to the vesicle counterpart. And again, you can see the decrease in the error bar that is there. So, this is a robust way for us to really look at the binding of the alpha-Synuclein to the surface. So then we move on to really trying to get at what is the physiological role of the alpha-Synuclein when bound to the surface. And because of the higher scattering density, we have done sort of either biophysics technique, but one thing that we want to look at is really to use X-ray to get at the interactions and to see if we can if that can reveal what is the physiological function of the normal state of the alpha-synuclein versus the disease state of the alpha-synuclein. And we choose to go and look at it in terms of using X-ray photon correlation spectroscopy to allow us to look at the effective entity that is diffusing in the milieu when we have the absence of alpha-synuclein binding. So we just have the SSLB versus as we dial in increasing amount of protein into the system, and hence changing the lipid-protein ratio of what is in the system to see how it affects the state of the structure of these combination of lipids and alpha-synuclein. So let me explain a little bit about this curve. What is plotted here is a series of experiments looking at the correlation as a function of the time delay. So this is sort of, with the X-ray beam focus at the spot and really looking at the residence time, or, the correlation between over time as to how long this system sort of linger into the focal spot. And so the different curve represent the different amount of protein that is given to a fixed amount of lipid or lipid entity, which is coating on the SSLB in this case. And so the black one is the case when you just have the SSLB without any alpha-synuclein. And as you're dialing in the alpha-synuclein, you actually see the shift or the faster decay of the correlation curve as a function of time, suggesting that the diffusion is changes. The diffusion time through this volume is actually changing as you increase the alpha-synuclein to lipid ratio. And as shown in here, the correlation is given by this equation that is here. So the fact that as you're moving from black to red, so at the end you have 1 protein to 40 lipid ratio. And you can also see that even going from the green curve, which is 1 to 107, 1 protein to 107 lipid, you already see that this curve are stacking on top of each other, and we'll analyze to see what that means. So this just say that the curve are collapsing over there. So if I were to have a monodispersed, non-interacting colloidal sphere that is in the system, the delta G2 that is shown here follow this form that is shown here. So it has a exponential decay with the tau given by the diffusivity and the wave factor Q that is given there. However, the experimental fit give us not that equation, but give us a stretch exponential, which is shown in the equation here. The stretch exponential suggests that there is a dispersion in the relaxation time. So again, if I were to plot the tau, which is given by this equation here, as a function of Q, these are the curve that I found, which is the same data as in the previous curve, as a function of having different lipid to protein or protein to lipid ratio, as shown by this here. If I were to analyze it to get back what the diffusivity is, the top graph here shows you that when I have the absence of alpha-synuclein, the entity that is diffusing through this volume behave with a diffusivity as if it is a much bigger entity because it's going much slower, and therefore has a longer correlation time. But as I increase the protein to lipid ratio, what I see is that this diffusivity actually increases, and it approaches the expectation value of that of a 70 nanometer spherical object just freely diffusing itself, which correspond to how, when you back out the beta, which is the portion for this stretch exponent, what you see is that this beta also approaches the value of 1, which means that as you increase the alpha-synuclein in the system, you eventually approach the free diffusion of the 70 nanometers colloidal spheres in this system. So that's sort of the upshot of what the XPCS measurements give us. So that gives us a dynamic picture of how the interactions of the alpha-synuclein with the SSLB give rise to a change of the effective diffusing entity size as it going through this volume. And now we turn to SAXS to help us understand how the presence of alpha-synuclein has on the lipid membrane itself. And so what are shown here, again, the color coding is similar as before with increasing protein to lipid ratio as you go to black, from black to red. And what we see here is that, what is shown on the right is sort of, what you would expect when you have just a monodispersed vesicle, monodispersed colloidal particles that is there. And so what the X-ray data shows is that there seems to be a peak that is shown here. And this turns out to be a correlation peak at that particular Q value. And this correlation peak is actually consistent with having nearest neighbor distance that is due to the fact that the sphere and the sphere are actually next to each other. However, and this happens when you have no protein present, which is the black curve, and with low protein concentration as you go down. But as you reach a certain protein level, you actually see the disappearance of this curve. And you can also see that this curve, that this correlation peak is being ameliorated as you started adding protein into the system up to a point that it just completely disappeared. So what this data suggests is that SSLB, in the absence of alpha-synuclein, is in an aggregated state, which correspond very well or corroborate well with the XPCS data that shows a much slower diffusivity and also a beta value that is not equal to 1. So you have this sort of dilation of the relaxation time scale that correspond to these aggregates. And so we can also use SAXS to actually probe how the alpha-synuclein affects the force response between the lipid. And so one image that one can put in mind is that if I were to have an adsorb alpha-synuclein to the surface with the N-terminus being in a altered, alpha helical state that is embedded into the membrane, but with the C-terminus still as an intrinsically disordered protein, it might sort of look like it is forming a brush that is mediating the interactions between the two systems. So in a way, if I can use force, also using osmotic stress to osmotic forces to actually force the system to push them together, how it would look like as I increasing the stress and as a function of different concentration of protein, or if I were to ameliorate the effects of the protein interaction. And so what is showing you here is that what kind of, so we're putting osmolyte into the system to force the SSLB to be experiencing this osmotic stress, so they would be sort of, flock together. And also as a function of time of adding a salt. And so if I were to just look at a particular vertical line at a given salt concentration, sorry, this is anyway, with a salt concentration, you would see a transition of the red dot to the black triangle from a aggregated versus a non-aggregated state. And this, what SAXS give us is looking at the correlation peak or their lack of, as you are increasing the osmotic pressure into the system, right? And what you see is that indeed, if you were to increase the osmotic pressure, even though at a value that if you don't have the osmotic pressure, you would have a certain transition point whereby you would see the aggregation versus the disaggregation state. What you see here is that as you increase the salt level, this transition actually changes as a function of the salt concentration. So what we have done with the picture in mind, that you would have the N-terminus being inserted into the synaptic vesicle with the C-terminus still as an intrinsically disordered protein, we want to ask whether if we were to chop up this part, whereby you won't have that intrinsically disordered protein that is floating onto the top of the surface, how would it affect the behavior? And what you find is that indeed, if you were to chop that off at the same, this is looking at 100 millimolar concentration of salt, instead of a high, you need a high pressure to force this transition between aggregation and disaggregation because of the lack of the repulsive interaction that is given rise by the presence of this N-terminus. Because I have removed it, this reduce the osmotic pressure that is necessary in order for that transition to occur, okay? So what I hope I've shown you in this first part of the talk is this one particular system whereby we're able to change the surface chemistry and using osmotic stress to actually drive assembly of a single full coverage of lipid layer on the silica nanoparticles, which have all these features that we have mentioned before. And with that, we were able to do various type of X-ray as well as biophysical measurements to allow us to understand what the mode of interactions between the nanoparticle and the alpha-Synuclein, and give rise to both dynamic and static data to allow us to get at the modality at which the interactions of this protein to the lipid and how that surface association give rise to a protective, repulsive interactions that confer steric stability of the whole system so that they won't aggregate. So this sort of gives you a sense of the type of problems that we're interested in. And many of the projects in the lab pertains to trying to really get at a molecular understanding of the lipid-protein interactions that is at hand. So I'm going to switch gear a little bit for the second part, just to give you a vignette of the type of work that we can now do to get at orientation, penetration depth of a peripheral binding protein when it is interacting with a membrane. And this is sort of using a combination of 3 different techniques to allow us to inform one another and to sharpen our understanding of the system. So,
okay, can I have a question?
Yes, of course.
Have you ever seen the correlation between that alpha helical part of the synucleins, packing them in a regular kind of aggregates?
Okay, so no, we haven't been able to sort of pack them at that high density, but also because in the absence of a lipid surface or some sort of hydrophilic interface, they would be in an intrinsically disordered form. So they would not pack in any ordered way. I
have to tell you that we clearly see the assembly, and we can see the correlation between helices when we are packing the alpha-synucleins paper in preparation to the hydrophobic surfaces.
Yeah, so if you have a surface that would allow it to go onto it, then you would see it. But in our case, we have a splay surface, right? And so the way how they organize themselves is not. So we have seen, for example, in a different system, which also in beta amyloid, we do see packing of this in the beta sheet as they are forming this beta sheet as it is interacting with the surface. I don't know if that addressed your question, but given the geometry that we have, we did not see with the alpha-Synuclein packing on the surface, we do not see any ordering.
Okay.
Okay. So in this case, I want to turn to just a little bit of the motivation of what we are looking at. In this particular case, we're looking at sort of the membrane asymmetry and what happened when you have a disease state of a cell membrane. So as you know, membrane keep its negative protein on the inner leaflet of the membrane for the cell, and with it not being exposed to the outside. And it's an active process, so it needs a flippase and floppase to make sure that it keep this asymmetry on the surface. And if you were to scramble it, then it would give rise to PS exposure of a otherwise normal cell. And so this PX exposure has been sort of tied to a series of different biological processes. And what we have been interested is the PS recognition process, and in particular of how PS recognition can actually be a way to signal downstream type of events in the case of apoptosis, which is leading of the death of the cell. What we need to do is to really remove those cells. And that falls into two proteins that we are looking at. One is the TIM family of protein, and the other one is the milfat globulin protein. And they have various sort of differences associated with them. But we're interested in the commonality of them interacting with a negatively charged head group that is PS in the case of our system, but it turns out to be a little bit more complicated than that. So just to give you a sense, we're looking at this transmembrane immunoglobulin mucin containing domain. So what it just say is that it has a binding part that is on the top. It has a mucin part that is sticking out and is anchored in a transmembrane type of fashion. And there are three types of TIMs for which the structures has been sort of identified that allow us with some starting point, which will become clear as I go into our methodology. And so in this particular case, they have a single pocket that is dependent on calcium for PS binding onto the surface. And there has been sort of different biological work trying to identify what is the real biological function of the various TIM, but I won't go through the details of which. Suffice it to say, TIM3 is one of the family members, and it is an important protein in the family because it has been identified as a target for cancer immunology, even though the exact function of what it is interacting with, how it's interacting with it, to what extent, and what is it sensitized to is not completely understood. But what we understand is that it does seek out for surfaces and bind, just like its cousins of TIM1 and TIM4, with a surface that is presenting a negatively charged phosphorylserine on the surface. So this is just to show you the three crystal structure of the murine TIM1, 3, and 4, and this is the binding pocket that we're talking about. So in the interest of time, I'm not going to sort of go through the biology, but I just want for this particular group to show the methodology in which how we sort of look at this type of peripheral binding protein interaction. Again, this doesn't have the full penetration like a transmembrane protein into the system. So the first thing that we want to use, want to look at is really the affinity and the level of binding and the fraction that is bound to the surface. And in a particular case on the binding pocket, there is actually a tryptophan, and a tryptophan fluorescence shift when it is in a hydrophilic environment versus a hydrophobic environment when it is inserted into the membrane. And by sort of monitoring the association of the protein to the lipid surface and looking at the corresponding change in the spectrum, we can discern what is the amount of a protein that is bound onto the surface. So the system that we use here is no longer nanoparticle, is a large molecular vesicle system. And what we look at is how, as we increase the PS mole fraction from 0 to 10 to 20 to 30%, how we see the bound fraction change. And what we ended up is really seeing this sigmoidal curve, and you can fit it to the Hill model that gives you a Hill coefficient of 2.3 something, that tells you that this type of binding actually is indicative of multiple PS interacting with the TIM, which is shown in this formula that is there. And so, you know, with the technique that we use, we were able to combine X-ray as well as molecular dynamic simulation, along with this fluorescence, tryptophan fluorescence spectroscopy, to get at the modality of the interaction into the membrane that is shown in this picture that is there. And so this is just showing you the raw data of how, as I introduce the protein in the presence of the calcium, and as I were to increase the calcium, how that increased both in the intensity of the spectrum, as well as the shift of the maximal wavelength of occurrence at that. And we analyze these data to get at a way of how the protein is interacting with the lipid and what is the mole fraction. So we can do various calculations and trying to get at the subtlety of the different binding. But what is curious is that I have shown you a X-ray crystal structure that has a single binding site. So the question is, why does the Hill coefficient is not 1, but it's a number that suggests multiple timing? In fact, it's a number that is greater than 2, so it would be associated with, on average, 3 PS lipid that is on top. So this, we turn to Grayson's X-ray reflectivity to really get at, trying to get at the orientation as well as depth of penetration of the protein to the lipid surface. And I guess with this crowd, I don't have to go through the detail of the technique, but it allows us to back out what is the electron density as well as the length of the tail group, the head group, and whatever associated protein that you would have on the system. So this is just sort of showing, you know, in the absence of the protein, you would have this. And when you put protein into the system, in the sense that you would have the PS that is there, so that the binding would occur, what you see is that there is this add layer of the protein that is present, and you can either remove the calcium with, or you can have a system in the absence of any charge. In both cases, you really don't see any signature of the protein that is at the head group. And so one can go through the data analysis of the bound lipid on the surface, and this is based on some of the work that Mark Schlossman and his group has developed to looking at surface adsorbed and bound protein at the lipid interface. And what we do is that using the initial known crystal structure, even though that is just a freeze frame, but that would be our starting point. And what we do is that we sort of rotate it in all possible configuration of theta and phi, as shown in this figure. For each of the orientation, we can simulate what would have been by, sort of, chopping up the Z electron density. We can simulate what this orientation would give rise to a theoretical reflectivity curve, and compare this with the experimental curve, and do it for all the configuration with as high or as low a resolution that you want as you go through this different angle to compare with it with the experimental one. And what you find is that you will find some sort of a heat map by changing the theta and the phi angle, that you would get that there are hot spot or cool spot in this case, which has the lowest p-value, which has the highest probability of that is the correct orientation state. And so but because, the data does not preclude that there's degeneracy. So for example, in this case, we have two most probable spot. All of them by eye give very good fit to the proximity, to the closeness of the theoretical data, but they are indistinguishable. One would give rise to orientation A, and the other would give rise to orientation B. So we are lucky in the sense that we were able to distinguish that B is the incorrect orientation because of the fact that we are currently in this study looking at only the binding pocket. But one have to remember the binding pocket is actually tethered to the mucin domain part. And therefore, this orientation would not be a physiologically feasible one because the part that is interacting and is binding is actually the anchoring point to the mucin domain as shown in the figure on the right. So this allows us to sort of hone in on the orientation of the protein onto the surface, and the orientation is consistent with the various known features of this protein that is corroborated with the other techniques that we look at. Basically, it has a central PS head group level of the penetration of the hydrophobic loop, but the interfacial X-ray scattering does not really provide the X-ray atomistic details. So the third leg of this sort of self-consistent loop of informing the interactions comes in with empty simulation. And without going through the detail, we have reproduced various conditions whereby we look at, how the positioning of the PS is in relations to the protein and the binding pocket. We have looked at how far the hydrophobic residue, which is what gives us the tryptophan fluorescence shift that we're looking at. And we also sort of compare the simulated results with experimental results that we are looking. But what this orientation of the protein as it is situated itself at the membrane surface actually shows us the detail of the interaction point. And what is shown is that in addition to the binding pocket, which is important for the PS association, it also have 4 interfacial basic residue in close proximity to the head group of the lipid, giving rise to the additional electrostatic anchor and associating with the additional PS, which is inferred from the Hill coefficient of 2 point something of additional cooperativity effect of the interactions, giving rise to the stability of the associated state or the bound state of this protein to the system. Okay? So in order to just really show that, we do a mutation just to remove these 4 in a single site mutation case, whereby we take it out one at a time and replace it with by an aniline. And we actually with the one exception of this particular residue, number 48, we all see the effect of the change of the delta G when those charged residue that's in proximity of the membrane is responsible for stabilizing and enhancing the interaction of surface. So this, along with simulation, I showed you earlier, the simulation is the molecular dynamic simulation that allow us to look at a docked protein from the X-ray reflectivity data to see the validity over a time scale that we have of that. But one can imagine that that is a little bit different from the real experiment, right? In the real experiment, we have the film, we inject the protein into the subphase, and we let it seek for the surface, and then eventually found the bound state. But we don't have that luxury with a all-atom molecular dynamic simulation. So all we could do in those earlier simulation that I showed you is to use that as a starting point and see that whether that is stable or not, because we cannot seek the surface. So in collaboration with the Tokushak's group, we have used HMMM, so highly mobile membrane mimetic method, to really simulate the behavior of the protein as it is seeking for the surface. So I'm just going to rerun this one more time. Wow. The simulation with the protein actually in the water superface in this case, and looking at its docking onto the surface and recover the orientation that is very similar to what the X-ray orientation that we have obtained through an average X-ray reflectivity measurements that we have. Okay, so the one final point which is of interest is that, we talk about the PS is the one that is flipped out, but is that, very specific to PS, or is this interaction have some nuances that is looking for, is it just electrostatic or it has specific sort of lipid recognition? And what we have done is to look at, instead of just PS, we have looked at we have sort of titrated in other negatively charged haplo. In this case, this is with PG, with glycerol head group, and we find sort of, PG in the absence of any PS would still have some interaction with the system. And but what is very interesting is that if you have PA, phosphatidic acid, that interaction in the absence of PS is much more pronounced compared to the PG. And it turns out that PA is something that is actually produced and flipped out when a cell is undergoing apoptosis, but only a very minute amount of the PA, and this is sort of dialing in as with the total lipid content being anionic fixed to be 30, but I would dial in a little bit PA at a time. So when I am at 30% PS, that means I have no PA. If I dial in 0.5% of PA, I see that it really dropped the KD dramatically. Put in 1% of PA, drop it even further. And by the time I get to 2% PA, I am pretty much at the lowest KD that I would ever get. So it is very interesting how nature can actually use this high sensitivity, and I can go on forever we have spent, the last couple of years trying to understand what this PA sensitivity is, but that sensitivity gives rise to a complete retooling of the affinity of this particular protein to the surface and making it a competitive cousin in the family of TIM protein when the condition is correct to outperform other cousins for the binding so that it would have important downstream event. So this is very quick, I understand, but I just want to give you an example of the TIM protein, different members of which have different binding characteristics, and we were able to use this combination of tryptophan fluorescence, X-ray reflectivity, and molecular dynamic simulation to allow us how does the membrane context affect the sensitivity of the binding of the TIM protein to the surface. And this allow us to look at this protein, but I argue that this is a much broader and generalized statement that can be applied to all peripheral binding protein. So with that, I'll stop, and the credit where it belongs. The first part of the work is really the work of Peter Chung, who would be departing in, sadly, 6 days to start his faculty position at USC, and Luke Huang, who has since graduated, and Alexandra, who was an undergrad on this project, who has also graduated. The second project is really the work started by Greg Tejan, who is now a professor at Yale, but with work that is Dan Kerr, which I forgot to highlight the color. And the work that I show you, that I didn't show you, has to do with Tiffany work, which is with the MFG8 protein. So with that, I will stop and thank you for your attention, and my collaborators are listed down here. Thank you.